We're going to finish talking about some of the methods used to measure microbial growth. So why would we want to measure microbial growth? So it's really important in medicine to know, for example, how many bacteria are in a urine sample, because that'll give us an idea of how severe the infection is. And then we also want to know, for example, the number of bacteria in drinking water or in a food sample, and that'll give us an idea of how severe the contamination is. We can measure microbial growth by either direct or indirect methods, so we'll go through these. So these are the direct ways to measure microbial growth, and we'll start with the direct microscopic cell count. So we can use the Petroff-Hauser counting chamber, which is just a special microscope slide. So there's a counting chamber underneath here, and there's two little slits on the side where you put your liquid sample in. And then when you look under the microscope, there's a grid, and then you can count the number of cells that you see in that grid. So the advantage of using a Petroff-Hauser counting chamber is that it's really quick to get your results. So you don't have to culture the bacteria, which usually takes 24 hours to get results. So you can count and get your results like within a couple of minutes. So that's really nice. And then the other advantage is that it's really inexpensive to use one of these. So these are reusable. You can just wash them and then use them over and over again. The disadvantages to using a Petroff-Hauser counting chamber, the first one is that you can't tell when you're counting if the bacterial cells are living or dead. And so you're going to be counting both living and dead cells, which we really don't care about the dead bacterial cells because they're not going to cause a problem. We only care about the living ones. So that'll kind of put your counts off a little bit if you are counting both living and dead cells. So that would be a disadvantage. And then the second disadvantage is that if you have a small population size of the bacteria, then the counts can be inaccurate. So this is how the Petroff-Hauser counting chamber works. So when you look under the microscope, you would see one of these large squares, and you would just count the number of bacteria that you see in one of these large squares. So for this one, we have 14 bacterial cells, and we already know the volume of fluid that is held in one of these large squares. And so that would be 1.25 millionth of a milliliter. So if you have 14 bacterial cells, you just multiply that by 1.25 million. And so you'd end up with 17.5 million bacterial cells per milliliter of sample. I'm not going to ask you to do any calculations with a Petroff-Hauser counting chamber, but that's just how it works. And then the Coulter Counter, this is just an electronic device to count cells, and you put this into your sample. There's a little pump that will kind of suction the cells through a tiny little hole. The hole is small enough that only one cell will go through the hole at once. And then there's an electric current that's across the hole. So when the cell passes through the hole here, it'll break the current, And then the computer will count the break in the current and count that as one cell that's going through. So the advantage of using a Coulter counter is that it's very quick. So you're going to get your results right away. You don't have to culture the bacteria, which takes about 24 hours to get results. So you can get your results pretty immediately. The disadvantage, though, is that you can't distinguish between living and dead cells. So you're going to be counting both living and dead cells. So that would be a disadvantage. Another way to measure bacterial growth is to do a plate count. And the big advantage of doing a plate count is that it's only going to count living cells. And that's because you're going to be growing the bacteria in culture. So obviously only the living bacteria are going to grow. And we're only concerned about the living cells because they're the ones that will cause disease. And so we're not going to be counting the dead ones because they're not going to grow. The disadvantage is that it takes time to do one of these plate counts. So you're not going to get the results immediately. So it's going to take 24 hours to get results. And that's because you've got to incubate the plates for 24 hours and let the bacteria grow until you can actually see the colonies so that you can actually count them. When we do plate counts, we use serial dilutions, which are really important to understand. So we take 1 ml from our original sample, so we've got a urine sample from a patient, and then we put that 1 ml in 9 mils of culture broth, and that'll give us a 1 to 10 dilution. And then we take 1 ml of that and put it in 9 mils of broth, and that'll give us a 1 to 100 dilution. And then we just keep doing that all the way down here, and then we'll get a serial dilution and increase the dilution by a factor of 10 each time we do that. And then we'll take 1 ml of sample from each of these tubes, put that onto a plate, and then we'll culture that for 24 hours and then come back, and then we can count the number of colonies. And each colony here represents either started by a single bacterial cell or maybe by several bacterial cells. And so we'll report our count out as colony forming units instead of the number of bacterial cells, because we don't know if the colony was started by one bacterial cell or by several. And we want to choose a plate that has between 30 and 300 colonies. So we wouldn't want to choose this first plate because the colonies are too close together and they kind of merge together, and so it'd be hard to count them. There's just too many there. And the next one is kind of a lot. And then these last two, there's not enough colonies to count, and so it wouldn't be an accurate count. But the one here in the middle, that would be a good one to count. There's actually 54 colonies. And so then we would just take that number 54 and then just multiply that by the dilution factor, 1,000. And so you'd end up with 54,000 colony forming units per mil of sample. And then you would report that back to the clinician. So in order to get the sample from the tube onto the plate, there's two different methods that we could use. You could either use a pour plate method or a spread plate method. And the name tells you what you're going to be doing. So with the pour plate method, you would take your sample from the serial dilution tube and you would put it into some agar that's been warmed up, so it's been liquefied. Mix that together and then just pour it into the Petri dish, and then just kind of slosh it around, kind of spread it out, and then you would incubate that for 24 hours, and you can come back and count the colonies. And so it's just called the pour plate method because you just pour it out onto the plate. And then with the spread plate method, you would just take your sample and put it right onto some solidified agar that's already in the Petri dish. And then you take this L-shaped rod, glass rod, and then you would just spread your sample out onto the Petri dish. And then incubate that, come back 24 hours later, and then you count your colonies. And so you can see why it's called the spread plate method, because you end up just spreading the colonies out or spreading the sample out onto the Petri dish. Another way to count microbial growth would be filtration. So this is used in water treatment plants, and we use this in water samples where the number of bacteria is actually fairly small. So they would take a water sample, run it through a filter. The filter has really small holes in it where the bacteria will catch because they're too big to go through the filter. And then you would just take the filter, put it right onto the agar on the Petri dish, incubate that for 24 hours, and then come back, count the number of colonies, and then you report that out per however many mils of water that went through the filter. Okay, another method that can be used to get a count of bacteria would be the most probable number method. And this is also used in samples where bacterial numbers are low, so again in water and milk samples. And this just gives you a statistical estimation of the bacterial count. So what they do is they start off with 3 sets of tubes of culture media, and then they take different amounts of the water or milk sample, and then they just add that to the 3 sets of tubes. And then they'll incubate that for 24 hours and come back and see how many of the tubes have growth. And they're representing growth as the tube turning yellow. So what they'll do is they'll record, so like the first set of tubes, they saw that 5 of the tubes had growth. And then the next set of tubes, 3 of them had growth, and then the last set, only 1 tube had growth. And so they take those numbers, 5, 3, and 1, and then they go to this most probable number table, and they find that combination of numbers, so 5, 3, and 1. And the most probable number of bacteria that are in that milk or water sample is 110 bacteria per 100 mils. But it could range anywhere between 34 to 250 bacteria per 100 mils. So that's your 95% confidence limits. But the most probable number would be 110. So it's just a statistical estimation that could be used. And then just some indirect measurements of microbial growth. We'll take a look at spectrophotometry. Metabolic activity. This one you could measure, for example, oxygen consumption. And so if they're using lots of oxygen, you know that you have more cells versus another sample, if not as much oxygen is being used, then you have less number of cells. And then dry weight would be used to measure the amount of fungal cells. They would dry the fungi in an oven. Obviously that would kill them. And then they would weigh them, and then that would give you an idea of how many fungal cells are there. So I know you've used spectrophotometers in lab before. So remember, this machine will shine a light through the tube, and there's a detector on the other side that will detect how much light has gotten through the tube. And if your sample has a lot of bacteria in it, it will absorb the light, and then not as much light will hit the detector on the other side. And the machine will actually measure the absorbance, so how much of the light has been absorbed. And they actually have some standard curves that will allow you to measure the absorbance versus the number of bacterial cells. And so if you have a certain amount of absorbance that you've measured, then you can go over and say, okay, this is how many cells that I've got in my sample. So that's how the spectrophotometer works.